Mechanism of semi-conservative DNA replication. See, it is parental DNA strand. On this parental DNA strands, weak hydrogen bonds are identified by helicase enzyme. Weak hydrogen bonds means between adenine and thymine. Between adenine and thymine, two hydrogen bonds are present. The two hydrogen bonds are called weak hydrogen bonds. The weak hydrogen bonds are identified by helicase enzyme, and helicase enzyme identified the weak hydrogen bonds, and the two polynucleotide chains are leave from each other. This place is called replication origin. Replication origin. The replication origin look like a capital Y shaped structure. When the helicase enzyme helps in the cleavage of parental DNA, two long polynucleotide chains. The two long polynucleotide chains are act as a templates. On these templates, single strand binding proteins are attached. These are helps in they cannot turn into double helix structures. The templates cannot turn into double helix structure. Then after, on the replication origin, with the help of RNA primase, one RNA primer is synthesized. One RNA primer is synthesized on the replication origin. If RNA primer is synthesized on the replication origin, then with the help of DNA polymerase 3 enzyme, new copy of DNA is synthesized. New copy of DNA is synthesized from 5' to 3', from 5' to 3' direction without any disturbance. So this template is called a leading strand. But on the logging strand, RNA primers are repeatedly attached on the templates. And with the help of DNA polymerase 3, synthesize new DNA copy from the RNA primer fragment. And finally, the RNA primers are replaced with DNA with the help of DNA polymerase 1 enzyme. And finally, on the logging strand, fragments of new DNAs are identified. The fragments of new DNAs are called Okazaki fragments. These fragments are first identified on the logging strand, called Okazaki fragments. All Okazaki fragments are ligazed by the DNA ligase enzyme. Finally, from one parental DNA, two daughter DNAs are formed. From these two daughter DNAs, 50% of genetic material, 50% of DNAs are came from the parental DNA, and remaining the 50% of DNA synthesized with the help of DNA polymerase 1 and DNA polymerase 3 enzyme. On the leading strand, the new polynucleotide chain is synthesized with only DNA polymerase 3 enzyme. On the logging strand, the new polynucleotide DNA strand is synthesized by the DNA polymerase 1 enzyme as well as DNA polymerase 3 enzyme. Finally, the daughter DNAs are contain 50% of parental DNAs and 50% of daughter DNAs, newly synthesized DNAs. So this method is also called a semi-conservative method.